My name is Leslie Chung, and I'm the CEO of Imugene. I am very proud to bring you our company. It's my disclaimer. So first of all, Imugene currently has roughly 186 million in the bank. This is one of the highest cash balances of an ASX listed biotech company. And I'm even more proud that we spend largely 70% or more on our program. So we have 6 unique assets with 4 platform technologies, 4 scientific collaborations. We are addressing many solid tumors. However, with our recent acquisition, we are now in blood cancers. We operate under 6 FDA IND in the strictest criteria. And then we already have 2 supply agreements with major pharmaceutical companies. So our 4 unique platforms, and today, because of time, I'm going to talk about our allogeneic CAR T cellular therapy, azer-cel, our onCARlytics, and our CF33 oncolytic viruses. So azer-cel is a CD19-directed allo CAR T. We were able to acquire this. It's a first in class. It comes with a robust 84 patients worth of data. We also have 3 additional assets to target, as well as the registrational to market strategy, which is fast to market and potentially an accelerated approval. It could be the first FDA-approved allogeneic CAR T in the world. The drug has already been completed, and we have acquired the manufacturing facility along with a highly technically skilled and specialized workforce. So the main difference between autologous and allogeneic CAR Ts are this, in that autologous are really reliant on the patient's own T cells to be able to do the job. And so this gives you a limited patient access. And because there's a lengthy time in manufacturing, anywhere between 19 to 42 days, sometimes patient requires further bridging or further chemo before they can get their drug. It's highly a high manufacturing cost, and there's a huge variability in potency due to the variability in patients. Whereas our allogeneic, and especially our azer-cel, we have a broad patient access because we have healthy donors to provide the T cells that we engineer. It's available on demand and off the shelf, and there is no need for leukapheresis and no bridging required, as it is available when the patients are available. This has the opportunity for more efficient and cost-effective in terms of manufacturing. And because of that healthy donor cell engineer for potency and persistence, we're not relying on patient variability. So that 84 patients worth of data that we were able to acquire, 61 non-Hodgkin's lymphoma and 23 B-cell lymphoplastic leukemia. In lymphoma, we saw 58% overall response rate, and with leukemia, 61% overall response rate. But what is more impressive is that out of that 61 non-Hodgkin's lymphoma patients, 18 patients were auto CAR T relapse or auto CAR T failures. So those 18 patients got a whopping 83% overall response rate with 61% complete response, and importantly, that beautiful durability of response are greater or equal to that 6 months. So this is the reason why we acquired azer-cel, and we're going after a really difficult disease called diffuse large B-cell lymphoma. So it's an aggressive type of non-Hodgkin's lymphoma. Roughly about 80,000 patients in the US are diagnosed with non-Hodgkin's lymphoma, and out of that, anywhere between 30 to 33% becomes DLBCL. This is a high grade and fast growing and a huge high unmet need. And this is the disease that we will be going after. When you're diagnosed with DLBCL, first line of treatment is a combination of chemotherapy and steroids. About 60% of the patient population are cured with R-CHOP. However, those that progress either get a second line of high-dose chemotherapy with stem cell transplant, or more so with auto CD19 CAR Ts, moving into that second line. Once 60 to 70% of the patient population progress off of that auto CAR T, they do not have anything in the third line. So there is no real standard of care. And this is where we see opportunity and innovation with azer-cel. We know that roughly about 85% of the patients still maintain their CD19 biomarker, even though they progressed off of a CD19 therapeutics. And off the auto CAR T, we know anywhere between 60, now 70% of patients treated with auto CAR T relapse or fell off that line of therapy. So by 2025, the global CAR T relapse population will grow significantly as the sales of auto CAR Ts increase, just as these graphs tell you that DLBCL in first, second, and third line are all growing, as well as the sale of auto CAR T. Just 2023 alone, we're looking at 2.2 billion annual sales projected. This has grown exponentially since approval in 2017. Our clinical trials are currently running in major cancer centers treating auto CAR T diseases. So we know as soon as they fall off or progress off of auto CAR T, they can come on to our allogeneic azer-cel CAR T. So we have taken on the manufacturing facility as well as the research, and it's the significant amount of scientific know-how and knowledge that we were able to acquire. And we've already exceeded some of this timeline. So the batch that we will be taking into a potential pivotal or registrational study, that drug lot has already been released and approved by the FDA. We've also been able to transfer the IND to Imugene in a very short amount of time. So I'm going to introduce you quickly to our oncolytic viral therapy. There's only one oncolytic virus that is currently approved, and that's TVAC. And this was made by taking 9 different vaccinia virus strains, putting them in a vat, and then those that killed most tumors got to be called CF33, and we cloned those and put those through a high throughput screening process whereby they went against the National Cancer Institute line of 60 cancer lines, and it killed all 60 lines. So we know we have a very potent virus that we're taking into the clinic. What's so unique about our oncolytic viral therapy is that we have a human sodium iodine symporter that can be tracked in an imaging, so we know where our virus can go into the body. So in the simplest way, our oncolytic viruses love to infect only solid tumors and replicate in solid tumors, and then that whole endogenous immunity reacts and goes on to infect other cancer cells. And we have this in an industrial study where we're looking at intratumoral and intravenous dosing, as well as combination and IT with Pembro and combination in IV in Pembro. We're on cohort 4 of the monotherapy, and we are currently on cohort 2 of the combination. So we know that our oncolytic viruses only replicate in cancer cells, and our OnCARlytics, or our OnCAR19, replicates within a solid tumor, shuttles up or expresses CD19, and then when combined with the CD19 agent, it creates the endogenous T cell recruitment, the tumor cells lysis, and the virus is then released to impart more of that love onto solid tumors. So this is a program that we're putting together with Blincyto. We are very excited that we're moments away from enrolling into this first-in-class OnCARlytic study where we make your solid tumors into one CD19 target. By adding a CD19 directed target, you create a situation of obliterating and getting a really high response rate. So the combination with our allogeneic CAR T CD19 azer-cel and OnCARlytics is a reality. We now not only have a footprint in solid tumors, we have a huge footprint in blood cancers as well. One of the beauties of Imugene is that we have a diverse asset portfolio with multiple shots on goal. We have extremely experienced management team who have worked in solid tumors as well as allo CAR Ts. We have an ongoing clinical trials in diverse solid tumors as well as blood cancers now, and we have a robust cash runway to get us there. With the acquisition of azer-cel, it has expedited our corporate goal of company acquisition, our partnering with big pharma, our licensing technology separately, as well as developing and commercializing our products independently. I thank you for your time.